Well, thank you very much, Joe, for the introduction. And it's my great pleasure to talk to you about the microvascular health and the glycocalyx today. So today, what I want to do is focus on the microvascular system and explain a little bit more what's happening inside those smallest microvessels and what glycocalyx is and how important it is to keep those microvessels healthy. So when people look at the vascular system, they typically know the picture here on the left side of the slide. So they know if you look at the heart, the large arteries are taking the blood to the extremities and the organs, and the veins are taking the blood back to the heart. What many people do not know is that those large vessels only make up 1% of your vascular system. 99% of your vascular system and pretty much all of the contact between the blood vessels and the tissues is happening in the smallest blood vessels. So we call the microscopic capillaries. So these microscopic capillaries, they cover every part of your body. Any cell in your body, any cell in any organ is within microns distance of one of those microvessels. So when you zoom in with a big microscope and look more closely to those microvessels, well, this is what you can see. So this is one of those microscopic capillaries. When people started looking in more detail, like in this video, they started realizing that the red blood cells that you see flowing in the center of this capillary, they never touch the wall of the microvessel. And that was surprising because the vessels are only 5 microns in diameter, and the red blood cells, undeformed, are bigger than that. There were 6, 7, 8 microns in diameter. So they have to be deformed while they flow through these capillaries, and still they don't touch the wall. And then over the years, we have studied what's happening here on that interface between the red cells and the luminal surface of the vessel wall. And we now know that this space in between the red cells and the wall is not empty. It's not just filled with fluid. It's actually filled with a transparent compartment called glycocalyx. That's part of the endothelium. It's produced by the endothelium. It protects the vessel wall against damage, and it also mediates many, if not all, of the important functions of the endothelium that is required to keep your vessels healthy. Well, a few years after we got these kinds of microscopic videos in live tissues, we also developed specialized techniques to stain and image those structures within that space with an electron microscope. And this is shown in this image. And this paper was published in early 2000s and really made a difference in the understanding of people what's happening inside these microvessels. On the left, you see again a cross-section of one of those capillaries, and this is the myocardial tissue. And you see here on the outer edge is the endothelial lining, and on the surface, in between the surface of the vessel wall and where the red cells are flowing here in the center, you see all these bush-like structures. And on the right-hand side, you see a bigger magnification. On top would be where the red cells are flowing. And here's the endothelium lining the inner lumina of those microvessels and also the larger vessels. And now you can see the complex three-dimensional mesh of bush-like structures. And those structures are actually polysaccharides, like heparan sulfate and hyaluronic acid, produced by the endothelium and anchored into the membrane of the endothelium. So actually they make up part of the vessel wall. And these structures, although they appear to take up a lot of volume within that matrix, the solid structures only take up approximately 1 or 2% of the volume of that glycocalyx matrix. The other 98% is taken up by proteins that bind to the polysaccharides. And then the proteins attract and suck in a lot of fluids, building up a lot of hydrostatic pressure within that matrix and forming a very stable gel. So the volume of that glycocalyx matrix or gel that protects the vessel wall is mostly water. So in the conventional methods of preparing your samples for electron microscopy, one of the first steps before you try to make these images is to dehydrate your tissue. So take out all the water in your tissue samples. And you can imagine if you take out all the water of this matrix, it will completely collapse on the endothelium. And so the first images that people were collecting on glycocalyx, like in the 1960s, it was very, very small, only nanometer scale thin. And people didn't think it had any implications for endothelial function or blood volume in the capillaries. Now, with these improved techniques, we demonstrated and confirmed that these structures are extending up to microns in the lumen of the microvessels. So in a 5 micron vessel, If you have a 1 micron matrix on the surface of the vessel wall, that actually means in terms of volume that more than 2/3 of the volume in those small capillaries is occupied by this glycocalyx matrix. And only 1/3 of the volume is actually filled with blood under resting conditions. And a little later, I'll also show you that the volume that glycocalyx occupies is also very dynamic. So upon demand, it can allow blood volume to access more of that space. So it actually can control blood volume 3 or 4-fold instantly upon local demand when metabolism increases, for example, during exercise. So in this lecture, I will talk in more detail how we came about to develop imaging techniques and study the glycocalyx. And in the end of my lecture, I also will talk a little bit about a medical device called the GlycoCheck device that we've developed to study the glycocalyx and study the microvascular health in healthy people and people exposed to risk factors, as well as in patients that are suffering from a damaged vascular system and organ complications because of that. And in the end, I'll also talk about a therapeutic that we specifically developed to treat and optimize and improve the health of your glycocalyx. So the therapeutic is called endocalyx. It's composed of polysaccharides and glucosamine and antioxidants, and it's a patented formula designed to improve the health of your glycocalyx and improve the health of your microvascular space and hopefully prevent organ damage on the long term. Well, when I'm talking about glycocalyx and the years that we studied the different techniques to image and do experiments with glycocalyx, I do have to mention this person shown in this slide. His name is Brian Dooling. I had a great pleasure to work with him for many years when I was a young student in the 1980s and the 1990s. And Brian already at a very early stage realized that something's off in the microvascular space. At that time, people considered the microvessels to be glass tubes, nothing special happening inside. The whole volume of those glass tubes would be blood perfused, and there was no reason to think that anything would be happening on the luminal surface of those glass tubes. Well, Brian started working on some ideas to explain some observations in the early days, and I started working with Brian, in the late 1980s and early 1990s. One of the observations that started Brian to think about glycocalyx were these kinds of videos that people started collecting when they started using intraviolent microscopes in biological sciences. So these kinds of images were collected in the early 1970s, looking at live tissues with high magnifications and realizing if you look at the red cells flowing through the tiny microvessels and count the number of red blood cells in these tiny microvessels, that those vessels almost appear to be empty. So the volume fraction of the red blood cells in these tiny capillaries is extremely low. Only 5% or so of the volume of these vessels appears to be filled with red blood cells, which is significantly lower than the volume fraction of red blood cells in larger vessels. Because if you take a blood sample from a larger vessel and put it in a centrifuge, we know that 50% of the blood flowing in these larger vessels is composed of red blood cells. So there was a big discussion, why is there so few red blood cells in these tiny capillaries? Is the blood diluted, or is a lot of the volume inside these microvessels occupied by something else? Well, one of the studies that Brian designed was a brilliant study. It was done with small micropipettes to suck out the blood flowing through those capillaries. On the top left, you see an example of such a tiny micropipette. So he baffled these small glass tubes with very, very small openings at the end, and only a few micrometers openings, small enough to puncture an individual capillary and to collect the blood flowing through those individual capillaries. When he was collecting the blood that came out of those capillaries, and then he compared the red cell concentration with the red cell concentration in large vessels, he noticed that the blood flowing through those capillaries is actually the same blood as is flowing through the large vessels. So on the lower two panels, if you go from left to right, he started puncturing vessels, a little bit larger vessels, up to 80 to 100 microns, so the smaller arterioles, down to the smallest capillaries, and then from the capillaries to the small venules and the larger venules. And this, in the lower graph, he measured the discharge hematocrit. So that's the volume fraction of the red cells in the blood flowing out of the small microvessels. And he realized that, contrast to the visual estimate of only 5% of the volume in these vessels filled with red blood cells, that in the samples that he was taking from the capillaries, when spin it down in the centrifuge, still 50% the volume of the blood coming out of the capillaries is red blood cells. And in the top two panels, he compared the discharge hematocrit from the capillaries with the systemic hematocrit from the larger vessels, and you see they're equal. So the ratio of these two volume fractions is very close to 1, demonstrating that the blood is not diluted in these capillaries, and that something else must be causing the low volume available for red cells and plasma in these capillaries under resting conditions. Well, another observation that he made together with one of his students, Bruce Klitzman, already in the late 1970s, that although under resting conditions, when you see only few red blood cells in these capillaries and quite a few capillaries appear to be empty, that as soon as you start exercising and blood flow goes up, suddenly, within a second, many more red cells can fit into the same capillaries, and many more capillaries can be perfused with red blood cells. So there's a real dynamic change in the blood volume inside these capillaries on demand when you start exercising or you stimulate the tissue in any other way. Well, again, to test whether something was happening inside these vessels, allowing more blood volume to be there, he designed the following experiment, in which again he used these small glass micropipettes. And then he would puncture the feeding vessel, feeding the blood to those capillaries, and injected all kinds of enzymes to test if there's any biological compound inside these small capillaries that could be degraded by the enzyme, making more volume available for red cells. And indeed, he tested many different enzymes, and one of the enzymes that made a big difference in the volume inside the capillaries available for red blood cells was an enzyme called heparinase. The heparinase degrades the heparin sulfate glycosaminoglycans, again finding support for the possibility that some of these polysaccharides would be taking up a lot of volume in the microvessels, controlling blood content and possibly affecting endothelial function. Well, based on these findings, he came up with this hypothesis. If you have a cross-section of a capillary, as shown as this circle on the bottom, that in the center you would have the red blood cells flowing, and then on the outside of the red cell column, he hypothesized this hypothetical plasma layer known as the Glycocalyx. And that was the time pretty much when I entered the field and came work with Brian. So it was hypothesized there were some indications that there's something inside these microvessels controlling blood volume, but never anyone could make any positive picture, some evidence to support that claim. So when I started working with Brian, I started looking again at these tiny microvessels. And I injected all kinds of fluorescent dyes labeled to proteins flowing through those microvessels, trying to find evidence of these large proteins to stick on the luminal surface of the endothelium and occupying a lot of space. And I would be exposing the center of the vessel in the dotted area to high intensity fluorescent light, try to see evidence of protein sticking to the wall. And I tried to do that for over 2 years. I couldn't find any protein to stick to the wall until I realized if I looked at the same vessel again after 1 or 2 minutes, exposing it to the high intensity light, that suddenly within that time frame, the red cell concentration would increase significantly just inside that dotted area. Just turning on the light caused the number of red blood cells inside that space to double or triple. And looking back, if we hypothesize that the low red cell concentration in the first place was caused by an extensive glycocalyx, then realizing after 1 or 2 minutes exposing the vessels to high intensity light that the red cell concentration would double or triple, or the light itself must have damaged and destroyed glycocalyx, explaining why I wasn't able to see anything happening. And then again, when I went back to look at these sites in more detail, we realized that this is one of those capillary segments within the dotted area that before I would turn on the light, I realized there's only few red cells in that segment. In this case, there's 2 red blood cells inside. And when I started looking more closely, I realized that these red blood cells never touched the luminal surface of the endothelium. So the red cell column was more narrow compared to the inner anatomical diameter of the capillaries. Similarly, the fluorescently labeled proteins that were flowing with the red blood cells, if I measured the width of the red cell column, also the width of that flowing plasma column was significantly smaller than the inner anatomical diameter of the capillary. Also, the same as for the red cells, the large proteins that are flowing in the center of the vessel and do not have access directly to the luminal surface of the endothelium. Schematically, that was shown here in the graph on the right-hand side. So the black lines denote the position of the endothelium. Then we have a narrow width of flowing plasma proteins, a narrow width of the flowing red cells. And then once in a while, a much bigger white blood cell would come along that was so big and so stiff that actually could get close to the endothelium, indicating that these leukocytes can transiently compress whatever is in that space between the flowing blood column and the endothelium. Well, an example of the impact of a leukocyte flowing through a microvessel. So in the schematic on the right, we see that in a healthy vessel, if the black line denotes the endothelium, there is a narrow width of the flowing plasma column, also a narrow width of the flowing red blood cells. And then once in a while, a larger white blood cell or leukocyte would come along, fully occupying that space inside the vessel, suggesting that these white blood cells can transiently compress glycocalyx. Well, first I'm going to focus on the red cells and the width of the red cell column and see what happens if I expose the capillary to the intense light for just a couple of minutes. So here again on the left-hand side is the vessel. Before I turn on the light, only 2 red blood cells inside the capillary and a narrow width of the flowing plasma column. And the same vessel is shown on the right-hand side 1 or 2 minutes later after exposing the tissue to the high intense light to excite the fluorochromes. So after 1 or 2 minutes of high intensity light, damage to the vessel, glycocalyx is damaged, and now the red cell can now completely occupy the full anatomical diameter of the microvessel. So there's hardly any distance now between the edge of the red cell column and the luminal surface of the endothelium. They get much wider compared to the width they had before. And instead of 2 red blood cells, now suddenly 4 red blood cells fit in the same segment of the capillary. And again, the anatomical diameter of the vessel didn't change at all. Similarly, as for the red blood cells, after the light exposure of the vessels and damaging glycocalyx, now the width of the flowing plasma column significantly increased and fully occupies the full anatomical diameter of the capillary. Well, as I said before, in addition to the width of the flowing plasma column and the width of the red blood cells, we can also monitor the width of the leukocytes flowing through microvessels. And we realize that in small vessels, these larger leukocytes can transiently compress glycocalyx. And as a result, if you carefully trail what's happening behind the leukocyte, then the width of the flowing red cell column will transiently widen to occupy the full size of the vessels during the recovery phase of glycocalyx after passage of the leukocyte. So an example of that is shown here. This is a small arteriole with a diameter of approximately 10 to 12 microns. Here you see the red cell columns flowing, and you can appreciate it's difficult to estimate where the vessel wall endothelium is in this vessel because of the multiple cell lines bordering the red cell column. So if we stop the movie, you can get an idea on the width of the red cell column. And to better identify the position of the endothelium, we dye-labeled the endothelium, or we used mice with genetically altered endothelium to add dye inside the endothelium and to identify the position of the vessel wall. So if we switch the light to fluorescent light and see where the endothelium is, you can see that the real diameter, the anatomical diameter of the vessel, as shown by the fluorescent lining of the endothelium, is much wider than the width of the red cell column, similarly as in those capillaries. So the anatomical diameter is much wider than the width of the flowing red cell column going through those vessels. Now, if you're patient and wait for the passage of a leukocyte that transiently compresses glycocalyx, then you can see how much wider this red cell column can actually be if it would be able to occupy the full anatomical volume of the microvessel. And that's shown in this image. So in this image, on the top right, you see the back end of a leukocyte flowing through the vessel. So the blood is flowing from the lower left to the upper right, and then trailing the leukocyte. Shortly after the leukocyte compressed glycocalyx, you see that the width of the red cell column increased significantly. So if you compare that with the width just before, you can see it more than doubles the width of the red cell column, which means there's a 3 to 4-fold increase in the volume of blood in these microvessels when glycocalyx is compressed. Well, another method, again, to demonstrate and to support the claim that glycocalyx occupies a lot of space, shown here. Another technique to measure the volume of glycocalyx is to inject smaller plasma macromolecules like dextrans of medium size. The dextrans of 40 kilodaltons, if you put a label to it, they will be able to penetrate into glycocalyx. And this again is a small capillary. Now the positive stain is of the dextrans that accumulated in glycocalyx, again confirming glycocalyx is quite extensive and occupying a lot of volume in these microvessels. And again, with this technique, we were also able to measure the volume of glycocalyx with the dyes in humans and to see the impact of different challenges on the total volume of glycocalyx in the vascular space. These are just a few examples of what happens under different challenges with glycocalyx volume as measured with the small dyes. So we show that when exposed to an acute inflammation, like septic shock, the glycocalyx volume can decrease from 1 up to 2 liters, mostly confined to the microvascular space, to less than half a liter. There's an acute effect of smoking. So we had healthy students smoke a few cigarettes within an hour. And it was an acute effect of the smoking on glycocalyx volume, which recovered again after a few hours. And same for hypoglycemia. If we increase the glucose levels in the bloodstream to diabetic levels, we can see there is a negative impact on the hypoglycemia on glycocalyx volume. Now, with all these techniques that were developed over those years in the 1990s, in the early 2000s, we and many others were able to study not only the volume of glycocalyx, but also the functional implication of glycocalyx for endothelial function and microvascular health. So these two cartoons are actually summarizing many of those studies. And I won't go into detail to each individual study, but summarize, we know now that glycocalyx in a healthy vessel on the endothelium is crucial to sense the forward motion of the flowing blood, telling the endothelium to start producing nitric oxide. So these structures of glycocalyx, these polysaccharides that are anchored to the membrane, they act as amplifiers of the shear forces of the blood flowing over the endothelium. So healthy glycocalyx amplifies those forces about 100-fold, and that's essential to tell the endothelium to wake up and to start producing nitric oxide and to dilate the microvessels. At the same time, we know that a healthy glycocalyx is essential for the binding of antioxidant enzymes like superoxide dismutase. So in addition to stimulating endothelial nitric oxide production, a healthy glycocalyx is also very important to capture and to take away some of the oxygen radicals that are being produced by the vessel wall under different conditions. Well, in addition to the sensing of the blood and the activation of the nitric oxide synthase, we know that the tight nature of the polysaccharides in glycocalyx is a very important part of the endothelial permeability barrier. Prevents the leakage of plasma proteins like albumin and also the leakage of larger molecules like cholesterol through the vessel wall. Furthermore, we know that this extensive glycocalyx, which can be up to a couple of microns long, physically shields the endothelium and the adhesion receptors that are presented on the endothelium from interacting directly with other cells like blood platelets and leukocytes. So a healthy glycocalyx prevents the contact of platelets to adhesion molecules and prevents the contact of monocytes and leukocytes to the endothelial adhesion molecules. So it controls platelet adhesion and it controls the local inflammatory response. While when glycocalyx is compromised as a result of being exposed to challenges like high glucose levels, obesity, aging, hypertension, glycocalyx structures will be damaged. Now, red cells will get much more close to the endothelium. Proteins will start leaking. The nitric oxide production will be reduced, and platelets and leukocytes will start sticking to the endothelium, causing all kinds of clotting problems and local inflammatory problems. With this, I'd like to move on to the next part of the presentation and showing some information on the GlycoCheck system that we've developed to measure the health of the microcirculation and the glycocalyx in healthy individuals and in patients, and also later on show you a little bit of information on the early data that we collected on the effects of endocalyx, the therapeutic to repair glycocalyx, how that impacts also on microvascular health. So with that, I'm going to show you a little bit about the GlycoCheck device as shown on the left. So it's composed of a handheld microscopic camera that you can insert in your mouth to measure the red cells flowing through the microvasculature of the sublingual tissue. And then the software of the GlycoCheck system automatically counts the number of blood vessels and measures the health of glycocalyx and gives you a so-called microvascular health score. And on the right, again, is the endocalyx supplement that is designed to optimize the health of your glycocalyx GlycoCalyx, and we studied the impact of the treatments of glycocalyx microcirculation on endothelial function and organ health. So what I'd like to do right now is show you a little movie on the GlycoCheck device, so what it is and what we can measure with the GlycoCheck.
What's your health score? The score that identifies if you're at greater risk of the most troubling diseases and conditions. The score that signals that these diseases and conditions may be silently developing inside your body. The score that will help you understand how your body is alerting you with several early warning signs. For the first time, you're able to get an overall health score with a new, simple, and non-invasive test called GlycoCheck. It's a new patented technology, and the test can be easily performed in a healthcare practitioner's office. GlycoCheck gives you and your healthcare provider the one single number, a microvascular health score, that alerts you when trouble could be lurking deep inside your capillaries. The good news is that even if you have a poor score, you can easily improve your health. While you may look healthy on the outside, inside your microvascular system, a completely different situation could be unfolding. With the new discovery of a transparent lining in your capillaries called the Glycocalyx, your health could be declining because that lining may be breaking down and becoming leaky, which can contribute to the advancement of many diseases that develop over time. Aging, poor diet, lack of exercise, genetics, stress, inflammation, and smoking can cause the Glycocalyx to break down. In the past, there hasn't been a way to peer deep inside the microvascular system to observe the breakdown of the Glycocalyx. But now, for the first time with GlycoCheck, it's possible to see live movement of red blood cells as they travel through microvessels. GlycoCheck was created out of the need to measure the microvascular system by world-renowned researcher Dr. Hans Vink. Dr. Vink began his intense study of the Glycocalyx in 1989. Over the years, he has received many research grants and prestigious awards, all focused on the discovery and breakthrough understanding about the importance and clinical implications of the Glycocalyx. He has authored or contributed to hundreds of scholarly papers and reviews. GlycoCheck works by simply placing a video microscope under the tongue for a few minutes to video record, analyze, and objectively report a single overall microvascular health score. GlycoCheck counts the number of blood vessels and capillaries and measures red blood cell concentration. It also measures flow rate and if and how deeply the blood cells penetrate into the protective Glycocalyx gel lining, a sign of deterioration. GlycoCheck combines all these important measurements together to generate a single systemic microvascular health score. Lower scores indicate greater risk to the health of your organs. As the test is becoming more available, it can be administered in a healthcare practitioner's office. Each test includes a baseline score and video of the microvascular blood flow and tracks how, over time, the microvascular score declines with health deterioration or improves with therapy. Today, there are more than 85 ongoing clinical studies in more than 125 locations worldwide, such as Duke University, University of Pittsburgh Medical Center, VA Hospital of Salt Lake City, Children's Hospital in Philadelphia, the University of Oxford, Mayo Clinic, Leiden University Medical Center, Steno Diabetes Center, University of Exeter, and dozens more. These respected research centers are all using GlycoCheck to confirm that a damaged Glycocalyx contributes to several diseases and conditions, as well as the resulting complications from diabetes, stroke, septic shock, obesity, cardiovascular disease, hypertension, COVID-19, and other diseases. With GlycoCheck, now you can be tested and get the score that reveals the overall health of your microvascular system. With the results of your test, you will be in a better position to apply therapies and nutrition that can improve your microvascular health score.
So as shown in the movie, with the GlycoCheck camera, we get these kinds of videos live. So you can see the red cells flowing through the individual capillaries. And I'm going to explain a little bit of what's being measured from those movies by the GlycoCheck software. So the first step in the analysis of the microvasculature, the GlycoCheck software automatically identifies the position of all these microvessels. And along the length of all these microvessels, we define measurement sites. The one measurement site is a short segment of an individual capillary of about 10 microns long. And then we count the number of microvascular segments, and then we measure the diameter of all these segments, and we can truly zoom in on the smallest vessels. So we measure vessels with diameters between 4 and 25 microns in diameter, and the true capillaries are defined as the vessels with the smallest diameters, let's say between 4 and 6 microns in diameter. And one of the basic observations that we did with the GlycoCheck system is if you compare a healthy microvascular system as shown on top with a damaged microvascular system in a patient, that first of all, the number of capillaries goes down significantly. So on the top left here, in a healthy microvascular system at the low blood flow situation, you see that 2 capillaries are perfused with red blood cells, while in the patients, we see a significant drop in the number of blood perfused capillaries at the low flow situations. So the number of capillaries goes down from 2 to 1. While these data were confirmed by a recently completed clinical study on healthy individuals with obesity. So in this obesity study, we looked at close to 1,000 people, and based on the so-called Framingham risk score, which is a score that tells them how much risk they have in the next 10 years to develop a cardiovascular event, we've defined a low risk group with less than 10% risk, a medium risk group with risk between 10 and 20%, and a high risk group with people more than 20% risk. Well, in the graph on this slide, it shows you the numbers of capillaries relative to the low risk group. So 0 would be a normal level of capillaries. And on the horizontal axis, it shows you the diameters of the different microvessels. So on the left-hand side, you see the smallest capillaries between 4, 5, and 6 microns in diameter. And if you compare the number of smallest capillaries in the medium risk, in the orange, and the high risk in the red, with the reference level of low-risk individuals, you see already at this early stage of obesity, so these are still healthy individuals, you see already a 50% drop of the smallest 4 micron capillaries in the high-risk people that are at high risk because of their body size and the fact that they're smoking or they're getting older. This is already in healthy individuals way before they get organ complications. So that's an important parameter that the GlycoCheck system automatically measures, the number of smallest capillaries, and to identify a drop in the numbers of smallest capillaries in individuals at risk and being exposed to risk factors that damage your microvascular system. Well, another important observation that we did over the years, and which is being measured automatically by the GlycoCheck system, is that if you compare the numbers of capillaries in a low flow situation and in a high flow situation, that relative to the low flow situation, which is set at 100%, if you challenge the vascular system and blood flow goes up, and in this case, we increase blood flow by using nitroglycerin under the tongue. So in healthy individuals exposed to nitroglycerin, the blood flow goes up, but also the number of smallest capillaries that are blood perfused goes up in proportion to the blood flow increase. So here you see that the 5 micron capillaries, the number of blood perfused 5 micron capillaries increases about 2 to 3-fold in a healthy control group. While in patients, and these are 2 different cardiology patients, so the red line is for cardiology patients with obvious occlusions of large coronary arteries. The blue line is for cardiology patients with angina, chest pain due to microvascular problems. But both patient groups, they do not show any increase in the number of perfused capillaries upon an increase in blood flow. So in addition to the differences in numbers of capillaries at low flow between healthy and patients, we also see that in healthy individuals, you go from a low flow situation on the left to a high flow situation on the right. So the flow velocity of the red cells in the larger vessels increased from 80 microns per second to 120 microns per second, so a 50% increase in blood flow. There's also an increase in the number of capillaries from 2 to 3, so also a 50% increase in the number of blood perfused capillaries in proportion to the increase in blood flow flowing towards those capillaries. And as a result, although the blood flow increased 50%, if you look at the velocities of the red blood cells in the capillaries, they didn't change compared to the low flow situation. That's because that the 50% increase in blood flow through a 50% increase in numbers of capillaries, which keeps the velocity of the red cells very constant in the increased number of capillaries. While again, if we look at patients with a compromised microvascular system, in addition to the fact there's fewer capillaries under resting conditions, also when you increase blood flow from 50 to 100, you see there's no increase in the number of capillaries anymore. So they have a lower number of capillaries that are being perfused all the time, and they don't have a reserve capacity. And as a result, when blood flow goes up twofold, the only thing that happens at the capillary level is that the velocity of those red blood cells also goes up twofold. So the velocity in the capillaries goes up from 50 to 100, and the only impact is that the blood will spend less time in the capillaries when blood flow goes up there. So there's no benefit of increasing the blood flow. There's no increase in exchange conditions. And the only thing that happens is the blood flows faster in and out of the microvascular system in these patients. The next important parameter that's measured with the GlycoCheck system, obviously, is the health of Glycocalyx. And as shown before, that's done by monitoring the position of the red cells relative to the luminal surface of the endothelium. And in a healthy vessel, the red cells stay in the center and they never touch the endothelium, as shown here. So we measure the radial position of the red cells relative to the position of the endothelium, and they never get very close because they bump into that healthy protective gel matrix of the Glycocalyx. Now, when Glycocalyx is compromised, those red cells will just penetrate a little bit deeper into Glycocalyx, getting a little bit closer to the endothelium as a sign of Glycocalyx damage. So the depth of penetration of these red cells into Glycocalyx is taken as a measure for Glycocalyx damage. If we look at the differences between healthy individuals and patients, again, you see that the level of Glycocalyx damage is low in healthy individuals, and it increases slightly with age, but it increases much further if you look at critically ill patients in the ICU. For instance, if you look at sepsis patients, there's about a micron increase in the depth of penetration of red cells into Glycocalyx, which actually means that most of Glycocalyx is damaged and destroyed in these critically ill patients. Now, in addition to just measuring how deep the red cells can penetrate into Glycocalyx, we also realized that the differences in Glycocalyx damage between controls and patients, well, they can actually be more pronounced if you also take into account the flow dependency of how red cells interact with Glycocalyx. So that's shown in the middle graph. So on the horizontal axis is the blood flow going to the capillaries, and on the vertical axis is the level of penetration of red cells into Glycocalyx. And then the green dots are from healthy individuals, showing that in healthy individuals, if you take the flow dependency into account and you go to low flow areas, there is very little penetration of the red cells into Glycocalyx, while in patients, it's the other way around. At the lower flow sites, there's more and more damage of Glycocalyx and more penetration of red cells into Glycocalyx. So if you measure the flow dependency and take into account the differences in estimated Glycocalyx damage are much more pronounced, allowing us to get a much more sensitive estimate of the microvascular health score in these individuals. So if we take everything into account now, we measure the number of capillaries, we measure the flow dependency of the numbers of perfused capillaries, so the recruitment capacity, and we divide that by the estimated level of Glycocalyx damage. We've defined a so-called microvascular health score. So the healthier you are, the more capillaries you have, and the smaller Glycocalyx damage, the higher your microvascular health score. And typically in healthy individual, that score ranges between 4 and 8, up to 10 or higher. Well, we can compare the microvascular health score with people with a challenged microvascular system. These are sepsis patients in the ICU. You can see that their microvascular health obviously declines significantly. And the more damage they have, the more organs are being damaged, the more damage we also see in the microvasculature, telling us that a compromised microvascular system relates to the level of organ damage, and the more damage to your microvascular system, the more organs will be vulnerable for the dysfunction. Well, another study that we did recently was on COVID patients. These are a few graphs of that study on COVID patients. So we looked at COVID patients in the ICU. We had two subgroups of COVID patients, critically ill patients that needed mechanical ventilations to breathe, and a subset of COVID patients that were doing a little bit better, that did not need mechanical ventilation, they could breathe on their own. So the critically ill COVID patients are in the dark red. And in the middle graph, again, it shows you the loss of capillaries in those critically ill COVID patients compared to healthy controls. And I showed you before, with obesity, you start losing already about 40 to 50% of your capillaries. Now in the COVID patients, that was much more extreme. So the ones on mechanical ventilation, they lose up to 95% of their smallest capillaries. And that also explains why so many of their organs are at risk of being damaged also. So most of their microcirculation is wiped out. It's very difficult to take the nutrients to the tissue cells in the organs if you do not have any capillaries left. So again, if we calculate the microvascular score based on the number of capillaries and the level of glycocalyx damage, there's a very significant drop in the microvascular health score, especially in the critically ill COVID patients with the mechanical ventilators, and a very significant increase in the level of glycocalyx damage. And consistent with those findings, if you take blood samples and you measure the level of breakdown products of glycocalyx, so if you measure the circulating syndicants, which are heparin sulfates, they increase significantly in those individuals with the most level of glycocalyx damage. And also if you measure the breakdown levels of hyaluronic acid and other polysaccharide in glycocalyx, there's also a very significant increase in the circulating levels of damaged hyaluronic acid. If we have an individual with a compromised glycocalyx, can we repair that glycocalyx and microcirculation and help support their organ function with the endocalyx therapeutic? As I showed you before, it comes in bottles like these. The Endocalyx Therapeutic is a patented formula. It's a patent for composition and a method patent to treat the endothelial glycocalyx and to prove the health of your microvasculature. And briefly, the Endocalyx Therapeutic consists of three classes of compounds. There are certain specific polysaccharides from seaweed to repair glycocalyx. There is aminosucrose, mostly glucosamine, which is a rate-limiting substrate for the endothelium to produce more glycocalyx by itself. And there are specific antioxidants like superoxide dismutase from bitter melon that binds specifically to glycocalyx and helps to protect against further breakdown of your glycocalyx. A few slides to demonstrate what we can do with the endocalyx. So this is in an acute inflammatory challenge. If we break down glycocalyx with hyaluronidase, we see that within 30 minutes, there's a very significant increase in the level of leukocytes, inflammatory cells, to start sticking to the wall. So there's a 300% increase in the level of inflammation. And if you treat the subject with endocalyx, the endocalyx will help to preserve the protective properties of glycocalyx. And even in the presence of these inflammatory challenges, you can prevent endothelial damage and you can prevent the leukocytes to start sticking to the endothelium. Well, another finding that we did recently that was with the polysaccharides that we are using to help repair glycocalyx. In addition to just filling up the holes in glycocalyx, these enzymes also have another effect that helps to prevent damage to glycocalyx, and that's by binding an enzyme called heparinase. So the polysaccharide that we're using, a very specific fucoidan, looks very similar to the heparin sulfates that are endogenous in glycocalyx. And as a result, if you look at higher concentrations of the polysaccharide, it also will completely inhibit the heparinase enzyme activity, and thereby inhibit the breakdown effects of this enzyme that's increased in patients. So if you look at the patient's blood samples on the right, patients taking the fucoidan in the endocalyx supplement can very significantly lower their plasma levels of the damaging enzyme heparinase. And again, that contributes to preventing damage to your glycocalyx and keep your microvasculatures healthy. Well, just a few examples on in vitro studies on culture cells. So these are endothelial cells cultured in vitro using a technique called the atomic force microscope to measure the thickness of glycocalyx. So in healthy endothelial cells, you have a thick glycocalyx, and if you expose the endothelial cells to plasma from patients, like in kidney disease, you can significantly break down the dimension of your glycocalyx. If you use endocalyx to support the health of your endothelium, then even in the presence of the toxic plasma from the kidney patients, you can prevent the damage of your glycocalyx and have a normal glycocalyx and a normal healthy function of your endothelium. Similarly, if we look at the glycocalyx in mice, in their kidneys, the top line is a control healthy animal. And just focus on the center column, which is the slice of kidney, and kidney is stained with the lectins, which stain for glycocalyx. So the green dye shows microvessels with a healthy glycocalyx in your kidney. And if we look at diabetic mice, and again look at the kidney, then with diabetes, we see there's a lot of damage to the microvasculature and glycocalyx, so the green dye goes away. And then if we have the diabetic mice taking endocalyx for 8 weeks, as in the lower row, you can see even in the presence of diabetes, taking endocalyx helps them to preserve the health of their microvasculature and their glycocalyx. Well, one slide also to illustrate a recently completed clinical trial that we did with endocalyx on kidney patients. If you look at kidney patients and their microvasculature is severely compromised. On the left, you see there's only very few capillaries, the blood flow is very sluggish, and Glycocalyx is damaged, and they have a low microvascular score of 0.6. And after 3 or 4 months of treating the kidney patients with endocalyx, you can tell the difference. There's a huge increase in the numbers of microvessels. Blood flow is restored in the microvasculature, and their health score significantly goes up to 3.9. One or two more examples. This is on an individual with an open skin wound in the lower extremity. As you can see on the left, there was a severely open wound that didn't close, and the tissue, the rest of the lower limb was getting very dark and high risk of amputation. Also, this individual had a very high level of blood pressure, was hypertensive already for years, and did not respond to conventional antihypertensive treatment. So blood pressure was 200 over 110. Well, as soon as this individual started taking endocalyx, already within a few weeks, we saw that the color inside the open wound changed. So instead of being all whitish, the red color came back, reporting that blood flow was restored. And a few months later, the skin color normalized and the wound started closing. And then after about 4 to 5 months, the wound completely healed. But also what was surprising, if you look at the blood pressures in response to taking the endocalyx, after years of being hypertensive, within a few weeks, blood pressure normalized and stayed within normal range as long as this individual was taking the endocalyx supplement. So this also triggered us to start studying the impact of the endocalyx supplement and the healthy microvascular system on blood pressure control. And some information on the ongoing study that we are doing right now. If we look at 4 or 5 individuals that we tested so far with hypertension and look at their systolic blood pressures, within a few weeks, average systolic blood pressure dropped from 165 to within a normal range of about 125. Similarly, in a mouse model of aging, if we look at the impact of endocalyx on vascular and arterial nitric oxide production, we see if you take young mice and you compare that with older mice, in an older mice, there's a significant drop in the endothelial nitric oxide production. And as soon as we start taking endocalyx in the older mice, again, within 8 weeks, there's a significant restore of the level of arterial nitric oxide production, which also might contribute to that normalization of blood pressures as we discussed before. And the final slide is just to show you a healthy cross-section of healthy people that over time, taking endocalyx after 2, 3, or 4 months, look at their microvascular health and their capillary density, we see on average about a 50 to 60% increase in microvascular health after taking the endocalyx supplements only for a couple of months. The increase is still going on, so we expect in follow-ups of this study to find even better improvements of microvascular health if people take the endocalyx supplement much longer, and hopefully also thereby prevent endothelial dysfunction and organ complications. So with that, I would like to thank you again, Joe, for allowing me to present these data.